Biology, Chapter 12, Video 10, Theory, Mitochondrial Structure and Function. In eukaryotic organisms, what are eukaryotic organisms? Eukaryotic organisms are the organisms organisms with cells that contain membrane-bound nuclear materials. That is, a well-developed nucleus is present. The mitochondrion is the site of the Krebs cycle and the electron transport chain. So we discussed about the Krebs cycle and the electron transport chain. So both of these take place in the mitochondrion, either inside the mitochondrion membrane or the envelope, or in its matrix. These two processes take place there. So it is quite important. Mitochondria are rod-shaped or filamentous organelles of about 0.5 to 1 micrometer in diameter. Time-lapse photography shows that they are not rigid but can change their shape. Actually, there is a theory that says that mitochondria are actually microorganisms who have learned to live in collaboration with our cells. So they are not exactly structures that the human cells or eukaryotic cells have developed. They were initially microorganisms who have developed in a certain way to work in collaboration and exist inside host cells. So that is a theory. The number of mitochondria in a cell depends on its activity. For example, highly active mammalian liver cells contain about 1000 to 2000 mitochondria, occupying 20% of their cell volume. So that is quite a number. We are talking about a single cell. Single liver cell can contain between 1000 to 2000 mitochondria, occupying 20% of the cell volume. The structure of a mitochondrion is shown in figure 1.22. Page 16 and 12.13, right? So you will have to go and have a look at those. Let's go and have a look at them. So this is the mitochondrion enlarged by 20,000 times. So as you can see, the structure is not very fixed or rigid, right? So then we come to the current lesson, and this is also a mitochondrion. So now the structure of mitochondrion is like like a chloroplast, and each mitochondrion is surrounded by an envelope of two phospholipid membranes. The outer membrane is smooth, but the inner is much folded inwards to form cristae, singular crista. So as you can see, there are inward bound structures, and they are known as cristae, and they are extensions of the outer membrane. These cristae give the inner membrane a large total surface area. So that is obviously the function of it. Cristae in mitochondria from different types of cells show considerable variation. But in general, mitochondria from active cells have longer, more densely packed cristae than mitochondria from less active cells. So that means higher the activity, higher would be the amount and the concentration of the cristae. So that has, the cell surface, the surface area has an effect on how much energy has to be produced. The two membranes have different composition and properties. So we are talking about the inner membrane and the outer membrane. The outer membrane is relatively permeable to small molecules, whereas the inner membrane is less permeable. The inner membrane is studded with tiny spheres, about 9 nanometers in diameter, which are attached to the inner membrane by stalks. The spheres are the enzyme ATP synthase, as we saw earlier they are attached to these proteins, and they are so obviously these enzymes are proteins, so they are the site where ATP is produced. The inner membrane is the site of the electron transport chain and contains the proteins necessary for this. The space between the two membranes of this envelope usually has a lower pH than the matrix of the mitochondrion as a result of the protons that are released into the intermembrane space by the activity of the electron transport chain. So it has a lower pH, that means it is acidic. The matrix of the mitochondrion is the site of the link reaction and the Krebs cycle. The matrix is where link reaction and the Krebs cycle take place and contains the enzymes needed for these reactions. It also contains small 7-TS ribosomes and several identical copies of looped mitochondrial DNA. So the presence of these mitochondrial DNA is another factor that actually has a that gives us and gave birth to that theory of these mitochondria being separate or independent organisms. ATP is formed in the matrix by the activity of ATP synthase on the cristae. The energy for the production of ATP comes from the proton gradient between the intermembrane space and the matrix, and we discussed about that earlier as well. So what happens is the protons are removed into the intermembrane space, and when the concentration of these protons increase, there is a development of the potential. There is a high potential in the inner membrane in comparison to the matrix. So as a result, they start flowing back in through the channels. The ATP when they are flowing through, they actually release their energy, and that is what is used to produce ATP. The ATP can be used for all the energy requiring reactions of the cell, both inside and outside of the mitochondria. So that is basically the part. Before we go to the questions, let's write down a note on mitochondrial structure and function. Write down the topic mitochondrial structure and function. In eukaryotic organisms, within brackets, right? Organisms with well-developed nucleus, well-developed membrane-bound nucleus. Mitochondrion is the site of Krebs cycle and electron transport chain. Next point. Mitochondria are filamentous or rod-shaped organelles of between 0.5 to 1 micrometer diameter. Next point. They are present in large numbers in highly active cells. Example, between 1000 to 2000 in liver cells. Next point. Mitochondrion has two phospholipid membranes within brackets envelope. with the inner membrane having folded cristae. The inner membrane contains spheres of ATP synthase where ATP synthesis occurs. Due to the pH, due to the transfer of H plus into the intermembrane space, the pH there is lower than the matrix, lower than in the matrix. The pH there is lower than in the matrix. Next point, in the matrix, link reaction and Krebs cycle takes place. Link reaction and the Krebs cycle take place. And supporting enzymes (70S) ribosomes, and identical looped mitochondrial DNA are present. Right, so that's about it. I hope you understood. Please go through the note and then answer question number 12.4, 12.5, 12.6 and 12.7. If you have any questions, please raise them.